Alrighty, so we'll just begin now just to keep on time. This webinar will also be recorded and posted onto YouTube and our website as well. So keep an eye out for that and all of our other webinars. My name is Janet Down. I'm the Admissions Outreach Coordinator here at Keck Graduate Institute. And here with me today, I have Dr. Angelika Niemz, the Chair of Business and Bioengineering for the Henry E. Riggs School of Applied Life Sciences and the Arnold and Mabel Beckman Professor. Here we are today to give just a little webinar on the next generation sequencing. And so Dr. Niemz, I'm going to turn it over to you after we give the students a chance to introduce themselves. So I'll start with, I'm so sorry if I mispronounce your name, Leah.
Hi, that's correct pronunciation. Yeah, my name is Leah. I graduated from a Bachelor of Science last year in molecular biology and genetics, and I was hoping to learn a little bit about next generation sequencing.
Okay, perfect. Thank you. Thank you so much for being here. And then Lily, I don't know if you're back. Could you please introduce yourself?
Yes, I'm back. Thank you. I'm Lily. I graduated from UC San Diego and I studied molecular biology. I also got a certificate from UC San Diego Extension in applied bioinformatics. So I'm hoping to learn more about NGS. Thank you.
Well, congratulations.
So have you worked with next-gen sequencing data before?
I'm sorry?
Have you worked with next-gen sequencing data before?
In my class, we used like a tiny test data sample. So I wasn't able to play with like large data because my computer didn't have the capacity to process such large data. So just very little. Yeah.
Great. So you have a head start on some of the things we're going to talk about.
Well, thank you all for introducing yourself. Dr. Niemz, I'm going to turn it over to you.
All right. Okay, well, let's get started. Let me make sure I have the right screen. Okay, so sharing screen. Okay, so today it's my pleasure to tell you a little bit about next-gen sequencing in clinical diagnostics, which is one of my favorite topics. And so just to jump right in, DNA sequencing is basically reading the genetic code. And if you look at the cost of sequencing of a human genome, it has dropped from $100 million for the first human genome, and the fully loaded cost is actually more along the lines of 12 billion if you count everything that's associated with it. And now you can sequence a human genome for less than $1,000. So the thing that really made this possible was a technology shift. Prior to 2005, 2006, sequencing was done via Sanger sequencing, right? And you could update the platforms a little bit, but it really plateaued. And then in 2006, you had, like the Illumina platform and number of other platforms coming in, and then it really took off. So it was a technology shift that went from first generation sequencing, which is Sanger sequencing, to second generation, and the big 600-pound gorilla in the room is Illumina. So that's really clearly the market leader. We have some other companies in the race, IonTorn, and then there's third generation sequencing platforms such as the Oxford Nanopore and the PacBio. But the one we're going to mainly talk about is Illumina, right? Now, hot off the press, this came out on June 5th, 2022. So that's just a couple of days old. Ultima Genomics has actually claimed or released a statement that they can do a whole human genome for $100, right? So the technology is still evolving and more things are coming out that will increase the throughput and decrease the cost of sequencing. Okay. Next-gen sequencing can obviously be used in research, but it also has practical applications in clinical diagnostics, which include oncology, inherited disorders, non-invasive prenatal testing, and infectious disease diagnosis. So we're going to talk a little bit about all of those. And there are really two goals of this webinar. Thing one, we're going to talk a little bit about what is next-gen sequencing. And thing two, we're going to talk about how this is applied to clinical diagnostics, right? So starting with what is next-gen sequencing. The process of next-gen sequencing consists of these 4 steps. Thing one is you're going to have to get DNA from somewhere. So this could be a tissue sample, it could be blood, it could be, like some other, specimen. Then once you have the DNA, you have to prepare a sequencing library. Then you do the actual sequencing run, and then you have to analyze the data. Okay, so starting with the front end, we'll talk a little about the library preparation. So if you think about it, a human genome consists of 23 chromosomes, and the haploid human genome, which means just one set of copies, is about 3.3 gigabase pairs, right? Or billion base pairs, so 3.3 gigabases. Those 23 chromosomes are big. So it's not really, it's not really possible to handle them like that. So what you have to do before you actually can sequence a human genome, you have to break these big books up into smaller manageable books, right? So now what you end up with millions of these small books. And initially it's sort of a mess, right? They're all over the place. So think about it, how many fragments do you need? So you have 3.3 gigabases human genomic DNA, right? Times 2 because diploid. So you shear them into fragments that are 2 to 5 kilobases long. So that's about 1 million fragments per haploid genome. Now, the trick is you're not only going to sequence one copy of the genome, you're going to extract and shear multiple copies of the genome, right? So think of, you have multiple copies of these books and you break them all up and you break them up in different ways. So if there's one long story, you break it up into different fragments, and these books, these little books kind of overlap, right? So like one is chapter 20 through 25 and the other one is chapter 17 through 22. So they kind of overlap, which is important later on when you're doing the sequence assembly. And so what that really means is if you're looking at the genome, every single base shows up in a lot of different books, right? And so there's this concept of sequence coverage, which basically means how often does a base or a letter show up in different books, right? Now, 30x coverage is standard for whole genome sequencing, and you can identify inherited disorders with that, which means every single base, you have 30 different fragments or 30 different books where that base shows up. Now, if you want to use it in oncology, for prenatal testing or infectious disease diagnosis, you have to do deep sequencing, which is over 500 times coverage, right? So you have for each base, you have about on average 500 books where that base shows up. Okay, now interrupt me when there's any questions, because I'm going through this kind of fast. So if you guys get confused or so, just, like put something in the chat or just raise your hand or speak up. Is that okay? All right. All right. So moving on, now you've got those books and now you have to actually do the sequencing run. The way this works, so if you have a book or a fragment of about 2 to 5 kilobases, you're not actually going to read the whole thing. You're only going to read about 100 to 500 base pairs, or in some technologies, it's a little bit more. We're just talking second generation, which are the short read technologies. So in a second generation sequencing platform, you read about 100 to 500 bases on the front, and then some platforms such as Illumina, you're able to then cheat and also read the back of the book. If you're like me, I don't mind spoilers, so I read a little bit in the front and then I read a little bit in the back to figure out if I like the book, right? So this is called paired end reads. Now, what you do then is you have these overlapping fragments, and then later on you have to assemble to get to an actual genome. So you have to read a lot of books. So if you think again about the human genome is 3.3 gigabases and you do 30x coverage, right? So 3.3 times 30 is 100 gigabases, right? 100 billion bases, or 0.1 terabases. So just put that in the back of your mind. A human genome is about 0.1 terabases, right? Which is a lot. Now, if you have 100 base pair read lengths and paired end reads, so like you get 200 per read, if you take 100 gigabases divided by 200, you need to read about 500 million different fragments or 500 million books, right? That's a lot of books. Now, the way this works, so I'll use analogies a lot. So imagine for a moment, you're sitting in a huge classroom at, one of the big UC campuses, right? So you've got like 400 seats in that auditorium. Somebody sits in the middle and the back, and they have a book and they read, and I stand in the front. I can't hear them. So for this to work, what you actually need, you need 400 students or 500 students in the auditorium, and they all have the same book. And then they all start reading from the same book. And I stand in the front and I'm like, oh, yay. And now I can hear it. So they read a letter like A. So I write down A. And then I also write down how well I can hear that it is A, right? So you get the base call and you get a quality score, right? And then they continue reading. T, I write down a T. C, I write down a C, and so on and so forth, right? So everybody in the auditorium reads, and I write down what I hear. Okay, so but it's important that everybody has the same book, because if in that auditorium people had different books, then it would be complete gobbledygook and I wouldn't be able to understand anything, right? So for this to happen, this is where the library preparation comes in, because what you do is you take the genomic DNA, you shear it, but now you have to sort it, right? So you have to separate the fragments. So think of it this way. You have not just one auditorium, but you have 500 million auditoriums. You take a fragment, you put one fragment in each auditorium, and then you clonally amplify it. You make many, many, many copies of that same fragment, but they're all in different places, right? And then you can read and sequence the DNA. So the way this is done with Illumina is as follows. Illumina uses these modified bases, right? So when you do a sequencing run, this one is sequencing by synthesis, which means you have your fragment, you have a primer, and then you stick bases into, the thing like you would do normally when you're making, like doing PCR or something like that. Now, the trick here is, this thing has a block on the 3' end, which means you can only put one base in at a time, because you stick one in here over there at the 5' end, but then it's blocked here. And you have a fluorophore that fluoresces, right? Fluorophore, right? And what you do is you have 4 different colors. So you have A is red, T is green, C is blue, and G is yellow, right? So you can tell which base it is based on the color by which it lights up. So what you do is you have your different auditoriums. So these are the different clusters of clonally amplified fragments, right? And then you have your sequencing primer and you stick one base in. So on this auditorium or in this cluster, the base you stick in is a T, right? Because it's green. So you say, okay, this one, the first base is a T. Now what you do is now you remove the 3' block and you remove the fluorophore. So now it doesn't light up anymore. And now you can stick in the next base. And here over there, the next base you stuck in is an A. So now I say, okay, it's an A. And then what you do is you continue doing this, right? So you say here, this was a T, next round it's A, next round it's A, next round it's G, next round it's T. So by looking at the color that shows up for every cycle, I can say that this sequence is TAAGT. Now you see you have a whole bunch of other clusters, so you do this all at the same time. You read the books all at the same time in all the different classrooms. So this one is GCTGA, right? So this is why it's called massively parallel sequencing. And Illumina, in their newest version of the flow cell, they have these small wells that are arrayed. And so you can, fit things in like tightly clustered. So you literally have billions of these little wells. So imagine reading billions of these books all at the same time, right? And remember, before we said that for a human genome, you need about 500 million reads. Right? So if you can do billions of reads, you can actually do more than one human genome at the same time. So that's just crazy. Okay, so the total sequence output is the product of the read lengths and the reads per run, right? So how many letters I read overall depends on how much of the book I read in one classroom, right? Or auditorium. times how many auditoriums I have. And if you're looking at all the different platforms that are available, Sanger sequencing has a long read length, but it can only do 100 books at a time-ish, right? So that one is kind of low, versus some of the like the HiSeq and HiSeq X platforms that you have from Illumina, they can get over 1,000 gigabases. So this is over 1 terabase. So you get more than 1 terabase of sequence output per run. And if you're looking at this here, remember we said that the haploid human genome is 100 gigabases at 30x coverage, right? Or 0.1 terabases. So basically per run, it's more than 1 human genome. So it's just mind-blowing. Okay. So this is all good until you get to the data analysis. So the problem is, like, one of the huge bottlenecks is that you just get so much data and then you have to do something with it. So again, what you do, I'm just giving a very brief synopsis here because this gets really complicated and I'm just giving sort of a little primer. When I stand in the classroom and I listen to what is said, I write down the sequence and I write down the quality score, right? So this is the FASTQ, it's the sequence and the quality score. And then what I do is I have all these different books, right? So each one of them is a different book. So what I then do is I have a reference genome. So like the human genome has been sequenced before, so I kind of know where they go. So I can map those up, so I can take the reads and I can align them to the reference genome, like basically figure out where does this fit. And then I have to do some other data pre-processing, so you get the mapped reads. And then I have to do variant calling, basically saying, okay, I have my reference genome, I have those reads that are from, like, say, a patient with cancer. Where do I have, you know, single nucleotide variants or indels or some other genetic variation? So where do those books differ from my reference genome? And then I get a list of these variants, which is called the VCF file, which is variant call format file, right? So just to show you what that looks like. So the FASTQ files are based, I mean, there's a bunch of stuff in here. So each book gets 4 lines, but the important thing is, you see here, this is the sequence. And then down here is the quality score, which is in ASCII characters, but the quality score is like a number from 0 to like 40 or something like that. So usually, like, you want it to be higher than 30. Okay, so you have a lot of these because each 4-letter segment corresponds to 1 read or 1 book. So then you have to make sure that the quality is right? Because you see the quality score. So if it's like above 30, then it's great. So this one here is a good read, not so good, right? So you do data pre-processing, and if you see something like that, you're going to throw this out because something didn't work right here, right? So you do the quality check. And then you do this thing. So here, this is the sequence alignment, where here you have the reference genome at the top. And then each one of these here, these segments here, corresponds to a separate read. So I can say, okay, this thing here maps onto this segment of the reference genome, right? So this is the SAM, sequence alignment map, or, you know, BAM is binary alignment map. So this is basically the reads mapped to the reference genome. Now, the thing that's important is when you look here, this is all G. And then you see here, this is a C, this is a C, this is a C. So we have a polymorphism here, or a single nucleotide variant, where it goes like C to G or G to C, right? So what you can then do is you look across all of them and you do this variant calling, where I mean, this is the raw file, this is what it looks like. And then this is if you clean it up a little bit. So it basically tells you the chromosome, the position, what kind of, so this is single nucleotide variants, what the difference is, and so on and so forth. So this is just a list of all the variants that were found. And then what you can do is, so say, for example, you're looking at cancer. You have 100 patients that have colorectal cancer, and you have 100 patients that are normal. So now you look at the variants that you find in the normals, and you look at the variants that you find in the cancers. We all have variants in our genome, right? So we all have, you know, like 0.1% or something like that of single nucleotide variants that differentiate us from each other, right? So what you then do is you say, okay, here are some random variants that just pop up, right? They don't mean anything. But then you have some variants where you say, okay, all the cancer patients have this specific variant, right? And so then this is then associated with the phenotype, which in this case would be cancer, right? So what you can do is you can compile a list of all these variants, right? Mutations, insertions, deletions, stuff like that. And so we have this thing, for example, the catalog of somatic mutations in cancer, which is just a list of all the different variants that are found in all the different types of cancer. And you can then use that for clinical decision making. Okay, so now part 2. How are you gonna use all this in clinical diagnostics? We're going to talk a little bit about oncology, inherited disorders, non-invasive prenatal testing, and infectious disease diagnosis. So starting with oncology. There's a difference between somatic and germline mutations. So starting with germline mutations, so the germlines are basically the eggs in the sperm cell. So if you inherit a mutation that is in the egg or the sperm, every single cell in your body will display that mutation. And then when you have children, you can then pass it on to your children. So this is in inherited diseases. When you're looking at inherited diseases, you're looking at usually at germline mutations that are in every cell in the body and then can then be passed on to your offspring. Somatic mutations are like, okay, you are 40 years old or 50 years old, and then per chance you pick up a mutation in your liver that is oncogenic. So now you have a cancer that's growing in the liver that has a certain mutation, but the rest of your body cells doesn't, right? So this is called a somatic mutation, can lead to cancer, but it's typically not inherited, right? And so this is what you're looking for in oncology. Now in oncology or in general, you really have to think about, do I really need to sequence everything? Because there's a lot of stuff in the human genome. So when you're looking at this, the human genome, 1.1% of that actually codes for proteins. So these are the open reading frames, the things that actually encode for proteins. Then you have some RNA genes and regulatory sequences. And then you have a whole bunch of stuff that's considered to be quote unquote junk. It's not always junk. There's a lot of other good information in it, but it's not protein coding. And so what you can do, rather than sequence the whole genome, you only sequence the exome, which are basically the exons, which are expressed proteins. And so that then makes it a lot simpler. You lose information, but you also can streamline the process a lot. And then the other variation you can do is making it even simpler. Rather than sequencing the whole genome or the whole exome, you can do the thing that's called targeted sequencing. And the way this works is, say you have a certain number of genes that you know are related to cancer or an inherited disorder or antimicrobial resistance or something like that. So you take your genomic DNA, you fragment it, so you get all these different fragments, all these different books, and you say, okay, I'm only interested in these blue ones, right? So now you use a fishing rod. So you use these probes that are complementary to the books of interest. Right? Now these probes have a little label on them, it's called biotin. And basically what you can do is this thing here will bind to a streptavidin modified magnetic bead. So it's just a magnetic bead that can bind this thing here. So what you do is you fish out the fragments of interest, and then you pull this down and you wash away everything else, and then you only sequence that. So that's called targeted sequences. So you can actually fish out genes that you want to, and then just sequence that. Okay, now in oncology, there are a number of genes of interest. So this is just a nice infographic, right? So you have things like, BRAF, KRAS, EGFR, P53, so on and so forth. And I'm just going to give a couple of quick examples. So you may have heard of BRCA, right? Which is the breast cancer susceptibility gene, right? Then we also have, so this one here is increasing susceptibility. We have drug targets such as EGFR, and we have secondary effectors such as KRAS. So BRCA is basically a tumor suppressed. So BRCA is the breast cancer susceptibility gene. Normally, it's a tumor suppressor gene, so it repairs double-stranded DNA breaks, right? So if you have damage in your DNA, it fixes it back up, so you prevent cancer. Now, if you actually inherit mutations that make BRCA inactive, then you are at risk of early onset breast or ovarian cancer. So this is actually an example of an inherited mutation that leads to higher risk of cancer, right? So if you actually find out that someone has a mutation in BRCA, you want to do mammograms much earlier in their life, like really, make sure that you do intensive screening. And sometimes women will actually opt for mastectomy or, hysterectomy or something to prevent occurrence of cancer. Now, there's this thing called companion diagnostics. And the way this works is back in the days, you had a chemotherapeutic agent that only works in a small subset of patients, right? And then there are also patients that have a lot of adverse events, but you're like, okay, whatever, I'm just going to shotgun this thing. I'm going to treat everybody with the same thing, even though only 20% of the patients will actually respond and 5% of the patients will have severe adverse events. So now what we can do is we can actually sequence the genome or do other tests where we can predict, oh, you are going to respond to the therapy, you are not going to respond to the therapy, and you're going to have an adverse event. So we can personalize the treatment by saying, okay, only you will get this treatment and you won't, right? So it turns out that for BRCA, this is used for a companion diagnostic because I'm not going to go into all the gory details. So there's a couple of drugs that target a gene that actually, basically inhibits a gene that would fix DNA, right? Or, so the PARP inhibitor. So basically, if you have single-stranded breaks in the DNA and then you can't fix it, now you get these double-stranded breaks. Now, if you have an active BRCA, it would fix it here, so the drug would not work. If your BRCA is mutant, then you actually can't fix it, and then the cell is dead. So this is a little bit complicated. Don't worry about if you don't understand it fully. But the bottom line is you need to actually sequence BRCA to make a decision whether you should be getting this kind of drug, right? And so the first companion diagnostics based on sequencing were actually targeting BRCA. So there's a BRCA analysis by Myriad Genetics. This was actually Sanger sequencing. And then in 2017, Foundation Focus came out with the first next-gen sequencing test for a companion diagnostic targeting BRCA. And the way these tests work is basically you send a sample to Myriad Genetics or Foundation Focus. They then basically, analyze this thing, generate a report, and the report goes back to the clinician. Okay. So the other one I wanted to talk about is the epidermal growth factor receptor, or EGFR, right? So EGFR is expressed or upregulated in a lot of different types of cancers, like, breast, lung, colorectal, etc. And this thing has two important ends, the outside and the inside. So the outside is where the ligand binds, right? So epidermal growth factor, because it's the epidermal growth factor receptor. So the epidermal growth factor binds here. This thing dimerizes. And then you have an inside domain that's a tyrosine kinase domain, which basically phosphorylates different residues. And then it signals downstream, and then you basically get proliferation and cancer, right? So you have drugs that target EGFR to make it inactive, because if this thing fires off, you get cancer, right? So if you can inhibit, either you inhibit ligand binding or you inhibit the phosphorylation in here, you can prevent cancer, okay? So, however, there are mutations in EGFR that make the cancer, anti-cancer drugs either work better or not work, right? And so you have this EGFR tyrosine kinase domain, where again, this is a, several kilobase long stretch, and you have all these different mutations in these areas. So you have some that make the drugs work better, and you have some that make the drugs not work, right? Drug resistance. And so what you can do is you can sequence that, and then based on that, you can make treatment decisions. So the last example I wanted to show here is that you don't always just look at the drug target, because these things are all connected in pathways. So you need to make sure that you're looking at the context, the broader context. So if you think about here, you have EGFR, and EGFR signals to KRAS, and then KRAS signals downstream, and then you get cancer, right? So in a normal cell, KRAS only works if it gets a signal from EGFR, which means if you inhibit EGFR, then you get no signaling here, and then you get no cell proliferation. This is how the EGFR inhibitors work. Now, sometimes you have mutations in KRAS that basically turn it on all the time. So it fires all the time, regardless of whether it gets a signal from EGFR. So if you inhibit EGFR, there's no signal, but the mutant KRAS is on all the time, so you still get cell proliferation. So you wouldn't want to use an EGFR inhibitor if you have a mutant KRAS, because that would prevent the thing from working. So there are mutations in a number of different codons and a number of different exons on KRAS. And so originally we had these tests that were using PCR or qPCR, and they could look at like 7 mutations and a bunch of different codons. But the problem with that is you have KRAS, you have exons 2, 3, and 4, you have a number of codons, and then you also have another isozyme, you have NRAS, right? So here you have additional exons and you have additional mutation sites, right? So you see most of them, 40% are here, but you see there's quite a few that are in the other one. So if you only use this PCR assay that only looks at 7 mutations, you're going to miss a lot, right? Ta-da! Incoming next-gen sequencing. So Illumina came out with this Praxis Extend KRAS panel, which uses next-gen sequencing, and it basically looks at 56 specific mutations in KRAS and in NRAS, right? So it is sort of like it's giving you a more comprehensive picture and a more reliable answer as to whether your RAS is mutant always on and whether you should be using EGFR inhibitors to treat the cancer. Okay. Now you can use next-gen sequencing for oncology on steroids. So Foundation Focus came out with this Foundation 1 companion diagnostics assay. And this is basically going after breast, colorectal, ovarian, non-small cell lung cancer, and melanoma. And it looks at all those different genes, and it has all these different drugs that it basically can make treatment decisions for, right? So it looks at over 300 genes, over 25 exons, and it has a higher specificity and positive predictive value. This is just the list of the genes that they're going on. So when they're doing this fishing expedition, remember, like they're using these little probes to fish out the genes. They have probes in there that fish out all those genes. So they can sequence 300 different genes all at the same time. And so this is what it says here: hybrid capture next-gen sequencing based method. And this is just from the latest FDA database. So again, these are all the different drugs that require a test that will look for mutations in specific genes, right? So this is kind of like the one big thing that gets them all. Okay, moving on, we're going to talk a little bit about inherited disorders. So one example of an inherited disorder is cystic fibrosis. In cystic fibrosis, you have chronic inflammation and infection in the airways. And it's due to mutations in the cystic fibrosis conductance regulator, or cystic fibrosis transmembrane regulator, CFTR. And this thing is a chloride ion channel. And the way this works, usually in the airways, you pump chloride from the inside of the cell to the outside of the cell. Then the chloride, because it needs to be hydrated, it attracts water. And then it clears off the mucus that accumulates in the alveoli, right? Now, if your chloride channel isn't working, you don't get chloride exiting the cell, you have mucus buildup, right? So this leads to airway obstruction, and you also then get infections. So like Pseudomonas infections are very, very common in cystic fibrosis. And this is an autosomal recessive disorder, which means your parents are carriers, and the children, the offspring, has a 25% chance of actually having cystic fibrosis. Now, the trick is, in that gene, there are over 1,700 known mutations. They don't all mean something, right? So there's a subset of 139 mutations that are approved by the FDA to modulate treatment. Because it turns out that cystic fibrosis, it's not like one phenotype, right? It's not like one form. So what you can do is there's 6 different categories or 6 different classes, one of which where you don't make the receptor at all, one of which where you make the receptor, but it gets stuck in the endoplasmic reticulum, so it doesn't make it outside the cell, and then it gets degraded. One where you make it, it makes it to the surface, but it's not functional, or it has less function, or you make less of the receptor, or the receptor becomes unstable. And so these are all the different, or just example mutations that are associated with that. And then depending on what class of cystic fibrosis you have, you can make different treatment decisions, right? You would treat the patient with a different type of drug. And so Illumina actually came out with the MiSeq-Dx Cystic Fibrosis 139 variant assay that was FDA approved in 2013. So the MiSeq-Dx platform is FDA, well, not approved, FDA cleared. And then they have this assay kit that again is targeted sequencing that specifically goes after the cystic fibrosis transmembrane receptor, right? So this is an example for next-gen sequencing for inherited disorder. So here's another example. Neurodevelopmental disorders, they're reasonably common, right? So they affect over 3% of the children, but there are a lot of different types of diseases. These are all rare diseases, right? Usually they're Mendelian, so single gene, but a lot of them are undiagnosed, right? So you don't know what the origin is. And so what they've done is they did a study where they did whole genome and whole exome sequencing of 119 children with neurodevelopmental disorders of unknown genetic origin. And so what they found is that they had a pretty high rate of diagnosis, right? 73% if they were acutely ill, 40% if they were non-acute, right? And the trick here is a lot of these families, they go through a lot of different tests. So when you're looking at cost effectiveness, it's not just the cost of sequencing, it's the cost of what you would do if you didn't do the sequencing. So it turns out all the negative tests that they ran were about $20,000, $19,000. And the sequencing is, like $7,600, right? So like if the sequencing cost was $7,600, then it would be cost effective. But the important thing is also the sequencing at symptom onset. So the sequencing results were given 77 months earlier than the other results. So you can diagnose kids earlier and then you can treat them earlier, which makes a huge impact. And so there's a recommendation that children with neurodevelopmental disorders should be sequenced so you can speed up the diagnosis and ultimately also save money. Another area where this has been really important is in neonatal intensive care units, right, in NICUs. So they did this study where they were at 5 different hospitals. So they had 354 infants, and they did whole genome sequencing 15 days or 60 days after enrollment, right? And then what they found was in both study groups, whole genome sequence doubled the proportion of patients with a precision diagnosis and a change of clinical management. So by sequencing the babies, you can identify what the cause of the disease is, and you can change how you treat kids, which makes a huge impact. And so therefore, there are now recommendations that you should do whole genome sequencing of acutely ill infants with a suspected genetic condition, right? So if you have a kid in the NICU where you don't know what's going wrong, but you suspect that they have some kind of a genetic inherited disorder, you just sequence them. And there are now crazy advances in this field because Rady Children's Institute, for example, they can do ultra rapid whole genome sequencing where they can actually get sequencing done in 13 hours, right? The first human genome took 13 years, and now we're down to 13 hours, right? So that's just, stunning. Okay, and there's also a lot of different commercial players that are working in this area, like companies that do this kind of stuff. All right, moving forward to non-invasive prenatal testing. Now, one of the things that can and will go wrong in pregnancies are aneuploidies. Aneuploidies is basically when you have the wrong number of chromosomes. Like usually they're trisomies, right? Trisomy 21 is where you have 3 copies of chromosome 21. That's Down syndrome. Other common aneuploidies are trisomy 18, which is Edwards syndrome, trisomy 13, which is Patau syndrome, and then Turner syndrome is sort of like where you lose something rather than add something, where you miss all or part of one of the X chromosomes in females, right? And so what's important then is you're trying to understand whether your child has an aneuploidy during pregnancy, so you can make appropriate decisions, right? Because in some cases, children with Down syndrome actually have reasonable life expectancy and good quality of life. In other cases, you have serious malformations. A lot of the babies actually die, before they're born. And so it's important to kind of understand what's going on. Okay. Usually, when you're testing for aneuploidies, you do something like amniocentesis or chorionic villus sampling. So amniocentesis, you stick a needle into the amniotic sac, you pull out the fluid here. Chorionic villus sampling, you go, into the chorionic villus, which is part of the placenta. And these are invasive. And the trick is they can lead to miscarriages. So a lot of women, for example, when I was pregnant with my second child, they said, hey, do amniocentesis, because I was over 35. And I was like, nope, I'm not going to do it because I didn't want to risk having a miscarriage, right? So now what you can do is you can do this thing, it's called non-invasive prenatal testing, which is a blood test, right? So rather than actually having to stick a needle into your amniotic sac or into your, placenta, you can just take a blood sample, right? And then you can screen that. Now, the trick is it's a screening test only, right? So if your non-invasive prenatal test says you're fine, you're usually fine. If it says there's a problem, it means that you need to follow up. It's not a diagnosis. It doesn't for sure say that you actually have a genetic abnormality, right? So you have to follow up still with amniocentesis or chorionic villus sampling. So there are a number of companies that operate in this space, and there are two main approaches. One is massively parallel shotgun sequencing, where you just count reads, or you can do SNP genotyping, where you actually assign reads to, different, haplotypes. And this is just showing that, at the end of the day, with these types of approaches, if you have 200 pregnancies out of 100,000 with trisomy 21, which is Down syndrome, you can identify almost all of them. The other part that's important is having a low number of false positives or assay failures, because if you get a false positive, you have to follow up with amniocentesis, or if the assay fails. And so if these numbers are really low, then you can reduce the number of unnecessary procedures. So it has a positive impact. Okay. But wait, there's more. So rather than doing this while you're pregnant, you can actually do this before you actually have the embryo inside of the mom as part of in vitro fertilization. So what you do in vitro fertilization is you harvest eggs from the mom, you get sperm from the dad, you basically have the fertilization occur in a dish, and then you grow embryos. So you actually get a bunch of different embryos, and then you re-implant the embryos so that you then carry out the pregnancy, right? Now, the trick is, if you have parents who have a genetic disorder, wouldn't it be nice to figure out which embryos are okay and which embryos are diseased? So this is like crazy. This is genetic testing before the embryo even enters the mom. So the way this works is when you actually look at the early development, you have the 1 cell, 2 cell, 4 cell, 8 cells. So then you get the early morula and so on and so forth. And then the cells start differentiating, and then you get your blastocyst. And this is usually when you start doing, when you reintroduce the embryos. So what you do is you have your in vitro fertilized, embryos. You wait until you get to the 8 cell stage, then you harvest the cell, and then from that cell you can do sequencing, and then you can figure out what's going on with the embryo. And Natera is one company that does that. So basically what they do is they sequence the parents, they sequence DNA from the embryos, they figure out aneuploidies and other genetic abnormalities, then they pick the embryos that are healthy, and then they reimplant those back into the mom. Okay. So last but not least, infectious disease diagnosis. infectious diseases can be caused by a lot of different types of pathogens. You can have bacteria, you can have viruses, COVID, you can have fungi, right? And you can have parasites like, Plasmodium for malaria. And the important thing is when you have a patient with an infectious disease, you have to identify the pathogen because you need to know what causes the infection so you can appropriately treat it, right? So there's a big, who done it type thing. In clinical microbiology, so if you're in a hospital and you have an infection and your sample gets sent to the lab, they'll do things like gram staining and looking at it under a microscope. They will culture it, right? And then look at, how it grows on different media. And they can do biochemical methods to look at which bacteria have which enzymatic activities. And then there's some other methods such as immunoassays, molecular tests, mass spectrometry, and incoming next-gen sequencing. And the goal of next-gen sequencing is like one test to find them all, right? So it's sort of getting, again, comprehensive pathogen identification, and we'll talk about antimicrobial resistance in a bit. Now, again, you can do this different ways. You can do it by just looking at certain genes, or you can do it by looking at everything. So the first approach is targeted amplicon sequencing, which basically means you're targeting one specific gene. In this case, it's the bacterial gene for the 16S ribosome. So ribosome, again, you have the large to small subunit. For prokaryotes, right, bacteria, you have a 16S ribosomal RNA. So there's a gene for that, and that gene has conserved regions, constant regions, which are the blue ones, and variable regions, which are the pink ones. So if you have primers that target the conserved regions, you can then PCR amplify and make amplicons that have these variable regions. And then when you sequence them based on sequencing the variable regions, you can identify the species, right? So if you have a reference database of all the different bacteria, you align that. If they're over 97% similar, you say, hey, it's this bacterial species. And so you can do the pathogen identification. And you do this in clinical labs like the University of Washington Laboratory Medicine. You can send a sample to them, they do the analysis, and then they tell you what kind of bacterium you have, right? So that's targeted. The other version is metagenomic sequencing, where you basically just isolate all the genetic material, right? DNA or also RNA, if you're also looking at viruses. You do shotgun sequencing for all the DNA, and then you try from that to identify what the pathogen is, and so on and so forth. So there are a couple of companies that do this, right? Carius is mainly focused on blood. UCSF, like bloodstream infection. UCSF has done central nervous system infections, so they go after cerebrospinal fluid. They're also doing blood now. And DNA, they're doing mostly respiratory, like pneumonia and stuff like that. So the way this works, again, is at the clinic for Carius, so they're going off the blood, you take a sample, you send it into Carius, and they produce a result. Now, all that takes a bit. They say next day results, but the way it actually looks is because you have to ship the sample to them, so that's 24 hours. They sequence it in 29 hours, so it takes 52 hours until you get a report, right? Now, the red line is the pathogen ID that you can get with next-gen sequencing, and that black line is the conventional culture-based method. So you see here, it's still competitive timing-wise, and it also identifies more pathogens, right? So it found 50% of the cause of the infection rather than, 37 or however many that is, right? And this is just showing it in a different way. So the orange one is what you can get with conventional methods. The black one is what you can get with next-gen sequencing. And you see here, there's a number of different pathogens that were identified with next-gen sequencing that were not identified with conventional methods. So last, we have a problem. There's antimicrobial resistance, right? It's a real big threat now. And in the United States, there are about 23,000 deaths by antimicrobial resistance and over 2 million illnesses, and similar numbers in other regions. And there are a number of different bacteria that are really the superbugs, right? So we have vancomycin-resistant enterococci, Acinetobacter, methicillin-resistant Staph aureus, carbapenem-resistant Enterobacterialis, the extended spectrum beta-lactamase producers, and the multi-drug resistant Pseudomonas, right? So if you have one of those, you're in trouble. So, but wait, there's one way, I mean, there's a way of addressing that. Usually what you do is you grow these things in culture in the presence of an antibiotic. And if they don't grow, then they're susceptible. If they grow, then they're resistant, but that takes a while, right? And so you can hopefully get similar answers by sequencing. And so what they did is they have this comprehensive antimicrobial resistance database where they have basically 3,192 antimicrobial resistance resistant pathogens, no, 10,000, I'm messing this up here, but I mean, they have a lot of different, the punchline here is they have a lot of different resistant genes that are associated with antimicrobial resistance. Okay. So recently there's an assay that actually has hit the market, and this is so Illumina makes this is targeted sequencing in this case. So they make the kit that can fish out the genes, and then ID by DNA does the data analysis. So it basically targets 280 respiratory pathogens and over 1,200 antimicrobial resistance genes, right? So you can, through next-gen sequencing, say which is the pathogen, and also whether there are specific resistance genes present, which then would inform what specific antibiotic a physician should prescribe. Okay. And this is all I have. So with this, I thank you for your attention, and I would be happy to answer any questions.
Thank you so much, Dr. Niemz. If you would like to ask any questions, please feel free to put it in the chat or put it in, or just unmute yourself. Yeah.
Yeah.
Looks like you did a great job, Dr. Niemz. It was very informative. Thank you so much.
All right. Okay. Well, thank you for being here. And yeah, just let me know if you have further questions. You know, my contact information is available on the web, so you can reach out to me if you want.
All right, thank you so much. And then I put my email in the chat as well. So if you have any other questions or would like to know more about KGI, please feel free to reach out to me via email. I hope you all have a wonderful rest of your day. Dr. Niemz, again, thank you so much for your time. Oh, Lily asked, would the slides be available for us to review?
Sure, I can definitely do that. So I just put my email in here. And so what you can do is if you reach out to me, right, then I can send you the slides, and then, you know, I can also answer any more questions that you may have, right? So just reach out to me, aniemz@kgi.edu.
Yeah, thank you so much for attending today and for all of your time. And I appreciate you all. Good luck with everything, and I hope to see you soon.
All righty, take
Bye,
care.
everyone.
Okay, bye-bye.